All right, let's dive into a pretty amazing scientific detective story. We're going to piece together clues from these massive public data sets to figure out how a part of our own immune system can, well, go rogue and start actively mutating our DNA, leaving this really distinct fingerprint on cancer.
So let's start with the big question, the one that really drives this whole investigation. What if a major engine of cancer wasn't some outside toxin or just a random mistake, but something that's actually hardwired into our biology, something that's supposed to protect us?
To even begin to answer that, we have to start at the scene of the crime, the DNA inside a cancer cell. And our very first clue is this specific piece of evidence called a mutational fingerprint.
Okay, so think of it like this. Different culprits, like UV light from the sun or chemicals and tobacco smoke, they all damage DNA in their own unique way. They leave behind a characteristic pattern. And scientists can actually analyze these mutational signatures to figure out what caused a particular cancer. It's basically forensic science for our genome.
And this is where the plot really thickens. One of the most common fingerprints that scientists find across dozens of cancer types, we're talking everything from bladder to breast cancer, it doesn't point to an outside culprit at all. It points inward, to a family of enzymes called APOBECs. And get this, these are proteins our body uses to fight off viruses. So a key weapon in our immune system's arsenal is also a major source of cancer mutations. How is that even possible?
To figure that out, researchers had to zoom way in on the source, the specific neighborhood in our genome where these enzymes actually live.
It turns out that genes for the key players here, the APOBC3 family, they aren't just scattered randomly across our DNA. Nope, they're all packed together in this one tight little cluster on chromosome 22. Now, that kind of dense grouping is a big clue. It suggests they might all be controlled together, like a single unit.
And this is absolutely crucial. When they analyzed this region, they found something really telling. It's just loaded with somatic mutations. Those are the kind that pop up in tumors during a person's life. But it's almost completely clean of what we call germline risk variants, the inherited kind that might make someone predisposed to cancer. So what that tells us is that we're not looking at some pre-existing vulnerability here. We're looking at an active process that gets switched on inside the cancer itself.
Okay, so if it's an active process, something has to be controlling it, right? So the next step was to map out the complex genetic switchboard that's actually operating these APOBC genes.
Now, to understand this switchboard, you gotta know about something called enhancers. There are these fascinating little pieces of DNA. They aren't genes themselves, but they act like volume knobs or dimmer switches. They can reach across huge stretches of the genome to turn a gene's activity way up, or turn it way down.
And this, what you're seeing here, this isn't just a schematic. It's a map based on real data showing the physical 3D structure of our DNA. We can actually see how these enhancers, those bars up top, are physically looping through space to make contact with the APOBEC3 genes down below. It creates this incredibly complex, interconnected hub of control. And here is the kicker. The data showed that the enhancers in this hub that got mutated most often in cancer were also the most highly connected. I mean, they averaged over 5 connections to the APOBEC genes, compared to just over 1 for the enhancers that were mutated less often. This really suggests that these aren't just random mutations. They are hitting the most critical nodes in the entire control network. Okay, so we've got the genes. We've got this complex switchboard controlling them. And we know that switchboard is a hotspot for mutations. So the next question is pretty obvious, right? Who or what is flipping the switches?
To find that master regulator, scientists started looking for proteins that are known to be active in the immune system, proteins that might be physically binding to these very specific enhancer switches.
Well, the evidence points pretty clearly to one main suspect. When researchers analyzed several potential master switch proteins, one just stood out dramatically, NFATC3. Just look at that bar on the far right. While the others showed almost no connection, NFATC3 had an over 8-fold enrichment of its binding sites right at these enhancers. That result is so strong, there is no way it's just random chance.
And here you have it, the definitive proof. We are looking directly at the genome here, and those peaks show exactly where that NFATC3 protein is physically latched onto the DNA, smack dab on top of 3 of those critical enhancer switches. This is the molecular equivalent of finding the suspect's fingerprints all over the control panel.
So we followed this trail all the way from the mutational fingerprint to the gene cluster on chromosome 22 to this enhancer hub, and now, finally, to the master regulator NFATC3. It's time for the final step, connecting this entire chain of events directly to cancer.
This scatter plot right here is the smoking gun of the entire investigation. Every single dot is a different cancer type. And the key takeaway is that clear trend moving from the bottom left to the top right. The further to the right a cancer is, which means the more its APOBEC genes are turned on, the higher up it is, which means the more intense its mutational fingerprint. The link is just undeniable. The more active the genes are, the more damage they do.
So, let's put it all together. The story goes like this. We started with a mysterious mutational fingerprint that pointed right back at our own immune system. We tracked it to the APOBEC3 gene cluster. Then, we discovered that cluster is controlled by a whole panel of enhancers, which are themselves mutation hotspots. We identified the master regulator, NFATC3, at the controls. And finally, we showed that the more this entire pathway is switched on, the more mutational damage we actually see in cancer.
This whole discovery, pieced together from public data, it reveals how a totally normal biological process can be hijacked by cancer. It gives us a brand new set of switches and levers to study, and that leaves us with a really profound and honestly, a hopeful question. By understanding the intricate wiring of this internal mutator, can we one day learn to control it?